We continue along the hormone chapter and going back to the slide that ended the first recording. I want to give you more detail on this slide. We talked about the pancreas and how it's going to release either glucagon or insulin. So let's think of that. Let's say that you have high blood sugar. Let's say you just ate a huge meal. You just had a nice dinner, and that's going to, in the body, promote insulin release because you have all this sugar, right? You're breaking down that food, you're releasing glucose, and you want your cells to utilize that. You don't want it to just go to waste. So you want your cells to utilize that. So that high blood sugar is a stimulus to the pancreas. And what the pancreas will do is secrete insulin because it has a job to do now. The job that insulin is to use that sugar. If any cell in the body is needy and needs that sugar, that insulin is going to promote a change in that membrane that allows that cell to obtain that sugar, bring it inside the cell and utilize it. So here it's saying stimulates glucose uptake, right? It's the glucose uptake that's going to happen in these different cells. And it's in any tissue cell. And if it's going to go into tissue cells, it means I'm removing it from the blood. It's circulating around, right, this sugar. So if I add it to the cells, I'm going to, in turn, then remove it from the blood. If the cells don't need it, let's say the cells are full of sugar and they're not needing it, then another thing the insulin can do then is to make sure that it can stimulate the liver to form glycogen. What's that mean? It means store it. Let's store it. Okay, if none of these cells need it, then it makes sense to store it. Put it in the liver, and you put it in the liver in the form of a molecule called glycogen. Very big, long molecule with lots of glucose. So you store it. So two things that you'll do after your big meal. One is distribute it to the cells that need it, or if none of them need it, or just some of them need it, the rest is going to go into storage. So what's the outcome of that? Is that blood sugar is going to go down. We started with it high because you ate those too many burritos, and you brought it back down because your pancreas was working, the insulin was working, and the system was fine. Okay, let's see the opposite. What would happen if you're low blood sugar? You forgot to have breakfast, you ran out quickly, and then you didn't have lunch with you, and your blood sugar now is starting to drop because you haven't eaten. So then you are again going to stimulate the pancreas. But what you want to do here is to promote glucagon release. So not stimulate the pancreas, not to release insulin, but to release glucagon. What does glucagon do? Glucagon is going to stimulate the liver and have it release glycogen. Don't confuse those two, glucagon and glycogen. Glycogen is the form in which we store these sugars in the liver. Glucagon is the hormone. Glucagon then will stimulate the breakdown. Remember, we stored some of it here? So if we're going to store it, we need it now. Break it down, raise the blood sugar. So in that case, we started with low blood sugar. You hadn't eaten breakfast, you hadn't eaten lunch, and through this feedback mechanism, we raised the blood sugar. It's really important, this loop. Okay, what we want to look at more closely is how do cells of the pancreas actually release the insulin? How does that work? I mean, the mechanism of that. So we want to see at the cell level what's going on. It's specific cells of the pancreas because remember they can either release insulin or glucagon. So the ones that are actually releasing the insulin are the beta cells. The beta cells are the ones that are releasing insulin. So in our picture here we have the pancreas. If you were able to look at those cells, it's these specific cells that are the beta cells. And what they're going to do is, through the presence of glucose, because they're stimulated by glucose, they're going to release insulin. Okay, more detail coming. There was the same diagram again. This is a non-active beta cell. A beta cell is just sitting there. It's not responding to high glucose because there is no high glucose. It's just sitting there. You've had your meals, nothing's particularly high or low. So there's no need to release insulin if this is before a meal and you've eaten breakfast before that. So let's take it in steps. This, by the way, goes back to chapter 5. Remember, your book is set up in a way that the more simple mechanisms that you have to understand are in the earlier chapters. So you'll have to go back to chapter 5, figure 5.26. Let's take it in steps. If this were a cell, and let's say there's low glucose in the blood, this glucose is low, there's a different kind of glucose transporter. It's called the GLUT transporter. It's not the one we spoke of before. It's the CGLT, the one that will utilize sodium. No, this is one that's just going to transport glucose on its own. In the event of low glucose in the blood, then the metabolism within the cell is going to slow because I have less glucose coming in. If I have less glucose coming in at the cell level, the metabolism is going to be relatively low, and the manufacturing of ATP is going to be relatively low in this cell, which is fine. With a decrease of this ATP, we're going to go on to step 4 now. This potassium channels open. So remember the little ball, this is a gate, and this channel here is going to be open because of the low ATP. And then what happens? Well, if this doorway essentially is open, potassium can leak out. So it does. So potassium is leaking out here, all because metabolism is low, ATP is low. So the cell at resting membrane potential is not going to release any insulin. In fact, this voltage-gated calcium channel here that we're going to hear about a little bit later is closed. And that is important because by being closed, what it does is, see this insulin? Here's the insulin stored in this cell, but it's not releasing it. There's no insulin secretion. The insulin stays within the cell. Why? Because glucose level in the blood was low and essentially there was no need. So this will make more sense once you compare it to what does it look like when it's actually not resting. So let's go to that. This is an active beta cell of the pancreas. So now you've eaten. You ate those 3 burritos and you had a huge dessert, and now the glucose level in your blood is high. So now we better do something about releasing insulin. But let's take it one step at a time. Glucose is high. Sorry, how does that happen? Glucose is high. Here's my transporter that's going to bring some of that glucose. Now that it's high, it's going to bring it inside the cell through this GLUT transporter. Because it's bringing it in then, I'm breaking a lot of it down and I'm manufacturing a lot of ATP. So now ATP is high, unlike what it was before. If it's high, that channel that was leaking potassium before is now closed. If it's closed, then these sort of potassium positive ions are accumulating inside here. And they're accumulating because this is closed. And this charge of extra ions here is going to depolarize the membrane and make this calcium channel that before was sitting still doing nothing, it's going to make it open because of these positive ions that were not allowed to leak out, and they're accumulating. So this channel here is triggered by charge. So this charge is affecting the channel. What does a channel do? It opens. And what does that matter? Well, by opening, the calcium that was sitting on the outside of the cell here, sort of minding its own business, now has a place to come in. And calcium is going to flow inside the cell. And it's that calcium inside the cell that triggers these vesicles of insulin to migrate towards the edge here and actually release that insulin. Do you see where we got to? We got to from high glucose in the blood to a series of steps. ATP was high. What does that mean? Potassium channel closes. What does that mean? I've got a lot of ions. What does that mean? It opens the calcium channel. Why do I care? The calcium channel now is going to allow the exocytosis of insulin. That is an active beta cell of the pancreas. So it's going to be triggered always by these levels of glucose in the blood. So now that insulin is released, what happens next is that the skeletal muscle or an adipose cell is going to receive that glucose. So here's this glucose on a cell with an insulin receptor. And this is a resting skeletal muscle or adipose cell. And we have a glucose circulating. And in the absence of insulin, the glucose cannot come into the cell because this transporter, which is the GLUT4 transporter, is inside the cell instead of here. It's essentially not inserted. So what we need to happen is we need to get that GLUT transporter that's here, which is really a channel, bring it to the edge, and in that way allow glucose molecules to enter this particular cell that needs it. I'm going to stop this now and start the next recording with a picture of both of these side by side. In other words, one is in the absence of insulin, where glucose is unable to enter the cells that need food, and to the right, where insulin now signals the cell to do what? To insert that GLUT4 transporter. And by inserting it into the membrane, it's going to allow that glucose to enter the cell. Thanks for listening.